Welcome to this lecture on animal biotechnology. Today, we are going to explore one of the most powerful tools in modern genetics, the knockout mouse. If you have ever wondered how scientists figure out exactly what a specific gene does in a living body, you are in the right place. We will build our understanding from the ground up, looking at the concepts, the biology, and even the math behind creating these incredible animal models. Let us dive in. To start, let us imagine you are trying to understand how a complex car engine works, but you have no instruction manual. One of the most effective ways to figure out what a specific wire or belt does is to simply remove it and observe what stops working. If the headlights go out, you know that wire controls them. In biology, scientists use this exact same logic. Instead of car parts, they use living animals, usually mice. By intentionally disabling or knocking out a specific gene and observing what goes wrong, scientists can deduce the exact function of that gene in a healthy body. Now that we understand the basic logic, let us look at the formal definition. A knockout mouse is a genetically engineered laboratory mouse in which one or more of its natural genes have been intentionally inactivated or disrupted. This means the gene can no longer produce its normal functional protein. Because the protein is missing, the mouse is said to have that specific gene knocked out. These genetically modified mice are then bred and studied extensively. Researchers carefully observe how the absence of that specific protein affects the physical traits, behavior, and overall health of the mouse. Building on that definition, how exactly do scientists achieve this gene disruption? The inactivation is achieved by introducing an artificial piece of DNA into the cells. This artificial DNA swaps places with the normal gene through a natural cellular process called homologous recombination. When the artificial DNA inserts itself into the mouse's genome, it physically breaks the sequence of the target gene. This disruption renders the original gene completely non-functional. It is a highly precise biological cut-and-paste operation that ensures the target gene is permanently disabled in those cells. Let us visualize this mechanism with a diagram. At the top, you see the wild-type gene, which is the normal, functioning gene, containing exon 1, exon 2, and exon 3. Below it is the targeting vector, which is the artificial DNA introduced by scientists. Notice how the targeting vector has homologous sequences that match the ends of the wild-type gene, but the middle section contains a new neogene instead of exon 2. The dashed arrows show the crossing over event during homologous recombination. Finally, at the bottom, we see the resulting knocked-out gene. The normal exon 2 has been replaced by the neo sequence, breaking the original gene and rendering it non-functional. Having seen how the process works, you might wonder why we use mice instead of just studying human cells. The reality is that mice share roughly 99% of their protein-coding genes with humans. This makes them an incredibly accurate and reliable model for human biology. We cannot ethically knock out genes in humans to study complex diseases like cystic fibrosis, Alzheimer's disease, or cancer. Instead, we create knockout mice, missing the exact same genes that are mutated in human patients. This allows pharmaceutical companies to test new drugs and gene therapies safely before they are ever given to human volunteers. Now that we know why these mice are so important, let us discuss how we actually get a full knockout mouse. Creating them is a multi-step process, and the final step involves breeding. Because every mouse inherits 2 copies of every gene, one from the mother and one from the father, The first generation of engineered mice usually only has one knocked out copy. These mice are called heterozygous carriers. To get a true knockout mouse, we must breed two heterozygous mice together. The probability of their offspring inheriting the knocked out gene from both parents follows standard Mendelian genetics. This brings us to the mathematical representation of knockout breeding. We can express the expected number of homozygous knockout offspring using a simple probability formula. E equals N times P. Conceptually, this formula just takes the total number of offspring and multiplies it by the genetic probability of inheriting the trait. E represents the expected number of homozygous knockout mice in a given litter. N represents the total number of pups born in that specific litter. P represents the probability of a single pup inheriting the knocked out gene from both parents. According to Mendelian genetics, when two heterozygous parents are bred, P is exactly 0.25, or 25%. If the total number of pups, N, increases, the expected number of knockout mice, E, will also increase proportionally. Let us see this formula in action with a concrete example. A researcher breeds two heterozygous mice, and the female gives birth to a litter of 12 pups. We need to calculate the expected number of true homozygous knockout mice. We know our formula is E equals N times P. We substitute N with 12 because there are exactly 12 pups born. We substitute P with 0.25 because the cross of two heterozygotes yields a 25% chance for the offspring to inherit the knocked out allele from both parents. Multiplying 12 by 0.25 gives us 3. This means that out of the 12 pups born, the researcher can expect exactly 3 of them to be true homozygous knockout mice, completely lacking the functional gene. While breeding is the final step, we must also understand the immense difficulty of the very first step, engineering the cells. During the initial stage of creating a knockout mouse, scientists introduce artificial DNA into millions of embryonic stem cells. However, this biological process is incredibly difficult to achieve. Not every cell takes up the DNA, and among those that do, only a tiny fraction undergo successful homologous recombination. This creates a compounding difficulty where we are essentially calculating a fraction of a fraction. Understanding these low cellular efficiencies is crucial for planning the experiment and ensuring we start with enough cells to get a successful result. To quantify this compounding difficulty, we use another simple formula. S equals C times T times H. Conceptually, this formula applies successive probability filters to a starting population of cells. S represents the final number of successfully targeted stem cells. C is the initial number of embryonic stem cells used in the experiment. T is the decimal probability, or efficiency, that a cell successfully takes up the DNA vector. Finally, H is the decimal probability that the DNA correctly recombines with the target gene. Because T and H are both small fractions, multiplying them together shrinks the starting number C very quickly. If either efficiency T or H decreases, the final number of successful cells S will drop drastically. Let us apply this formula to a real laboratory scenario. A scientist introduces an artificial DNA vector into 2 million embryonic stem cells. The transfection efficiency is 10%, and the homologous recombination efficiency is 0.1%. We need to calculate the final number of successfully targeted cells. We start with our formula, S equals C times T times H. We substitute C with 2 million. We convert the 10% transfection efficiency into the decimal 0.10 for T. We convert the 0.1% recombination efficiency into the decimal 0.001 for H. Multiplying 2 million by 0.10 and then by 0.001 gives us 200. Physically, this means out of 2 million starting cells, only 200 successfully incorporated the knockout gene. We have covered a lot of ground today, so let us summarize the most important ideas. First, knockout mice are created by intentionally disrupting a specific gene to observe the resulting biological changes, allowing us to determine the gene's exact function. Second, this disruption is achieved at the cellular level through a precise process called homologous recombination, though it requires overcoming very low cellular efficiencies. Finally, by carefully breeding these engineered mice, scientists create perfect living models for studying human genetic diseases. These animals are absolutely indispensable for discovering new drugs and advancing the life-saving field of gene therapy. You now have a solid understanding of both the biology and the mathematics behind knockout mice.